Good morning, friends. Good morning, students. In today's class, I will discuss ketone bodies. Right. In this video, I will cover the introduction to ketone bodies. Then I will discuss ketogenesis, that is synthesis of ketone bodies. Then we'll discuss the reaction of ketone bodies. Right. And finally, I will cover the metabolism of ketone bodies in starvation and diabetes mellitus. Right. In the part 2 video of this series of ketone bodies, we will discuss the regulation. Right. So first of all, I will start with part 1, that is our introduction. Right. Now let's start with the introduction. So the name is ketone bodies, right? Ketone bodies, ketone, right? That means they contain a keto group, right? Right. So there are basically three ketone bodies. First is acetone, right? The second is acetoacetate, and the third is beta hydroxybutyrate, right? So remember one thing, that the acetone, the first one is, and the second one, acetoacetate, they are the true ketone bodies, right? But this beta hydroxybutyrate contains a hydroxy group, right? So it is not a true ketone bodies, right? And the properties are that they are water soluble, right? And they are energy yielding, and they are easily metabolized by the body. But the acetone is not metabolized, and it is excreted via lungs, right? And now I will start with ketogenesis. Keto means ketone bodies, and genesis means to synthesize, right? So the synthesis of ketone bodies is known as ketogenesis, right? So first of all, the where the site it is synthesized is the liver, where ketone bodies are synthesized in the body. Then the enzymes are present in mitochondrial matrix, right? And the precursor for the synthesis of ketone bodies is acetyl CoA, right? And acetyl CoA can be derived from, first of all, it can be derived from pyruvate, right? And moreover, it is also derived from beta oxidation of fatty acids, and it can also be derived from ketogenic amino acids, right? So now we'll start with the synthesis. First of all, the binding molecule from which the ketone bodies can be synthesized is acetyl CoA, right? The two molecules of acetyl CoA will combine in the presence of enzyme thiolase, right? And we'll get acetoacetyl CoA, right? This is the first step. We use two molecules of acetyl CoA and combine that with the enzyme thiolase, and we'll get acetoacetyl CoA, right? In the further step, we'll use another molecule of acetyl CoA, right? And we'll get beta methyl glutyryl CoA, right? And the enzyme is HMG CoA synthesis. And this beta methyl glutyryl CoA is also known as HMG CoA, right? Right. Now we got HMG CoA. Now in the next step, HMG CoA lyase will further break down HMG CoA into acetoacetate, right? This is our first ketone bodies that is produced, and one molecule acetyl CoA is removed in this reaction, right? And from acetoacetate we can form the ketone body known as acetone, that is excreted through lungs by spontaneous decarboxylation reaction, right? Here we require decarboxylation, right? And in other reaction, acetoacetate can be converted to the third ketone body, that is beta hydroxybutyrate by dehydrogenation reaction, and we convert NADH to NAD positive, and this requires NADH dehydrogenase enzyme, and we'll get beta hydroxybutyrate. So we synthesize all the three ketone bodies. First we synthesize the second, that is acetoacetate. From acetoacetate by spontaneous decarboxylation we synthesize the acetone, that is exhaled to the lungs, and the third, that is beta hydroxybutyrate, that is not a true ketone bodies, by dehydrogenation reaction. So right, so that finishes with our ketogenesis. Right, now we'll discuss the reactions of ketone. Now to start with the reaction of ketone body, first of all we have our beta hydroxy butyrate, that is our third ketone body that contains a hydroxy group, not a keto group, right? And this ketone body undergoes dehydrogenation reaction by beta hydroxy butyrate dehydrogenase, right? So NAD is converted to NADH and will get acetoacetate, right? And this acetoacetate is further activated to form acetoacetyl CoA, right? And this requires the enzyme thiophrase, and this coenzyme is generated by succinyl CoA, right? And this activated acetoacetyl CoA can undergo reaction by thiolase to form two molecules of acetyl CoA. So this is how the ketone bodies are degraded, and this enzyme thiophrase is totally absent in liver, right? So since this enzyme is absent in liver, so the ketone bodies are not utilized in the liver, right? So from beta hydroxybutyrate, it is converted to acetoacetate, then it is activated to acetoacetyl CoA, and by thiolase it is broken down to form two molecules of acetyl CoA by coenzyme ASH, right? Now the different tissues where we'll discuss, first is liver, then second is blood, and third is extrahepatic tissue, right? So first of all we have fatty acid, right? So fatty acid can undergo beta oxidation, right? And it will form the product that is acetyl CoA, right? And this acetyl CoA can enter the TCA cycle, first thing. And this acetyl CoA is also the precursor for the synthesis of ketone bodies, right? And when these ketone bodies will enter the circulation via blood, they are metabolized via kidney and lungs, right? From the kidney, they are excreted into the urine, also known as ketonuria, and from the lungs, acetone is excreted as acetone breath, right? And now coming on to the in extrahepatic tissue, the fatty acids, right? They will break down to form acetyl CoA, right? And acetyl CoA can enter the TCA cycle, right? From TCA cycle, it is further metabolized, and by glucose through glycolysis will form acetyl CoA, and acetyl CoA can further be metabolized by this pathway, and ketone bodies will also break down to form acetyl CoA by this reaction, so it is further metabolized. So that finishes with the reaction of ketone bodies. Now we'll discuss the utilization of ketone bodies. Right, now we'll start with the utilization of ketone bodies, right? First of all, the ketone bodies are water soluble, right? So they can be easily transported from liver to various tissues, right? Right. And moreover, these two principal ketone bodies, that is acetoacetate and beta hydroxybutyrate, they serve as important source of energy in peripheral tissue, right? So it can, whether it can be skeletal, muscle, right? Then can be cardiac tissue, and moreover in renal cortex. It serves as source of energy, right? And moreover, the tissues which lack mitochondria, that is our erythrocytes, since they lack mitochondria, so ketone bodies cannot be utilized in these cells, right? And moreover, the metabolism becomes important during starvation and diabetes mellitus, right? So during starvation, the brain is dependent on energy. The primary source is glucose, right? But during starvation, the ketone bodies meet 60 to 70% of energy requirement for the brain, right? So it is a defense mechanism for preventing the brain during time of energy deprivation, right? So that finishes with our utilization. Now we'll start with the metabolism in starvation and diabetes mellitus. Now we'll discuss the overproduction, right? So first of all, the normal level of ketone bodies in blood is 1 milligram per deciliter. That is normal level, right? And this during these normal levels, they cannot be detected by Rothera test, right? So when the overproduction take place, there is one delicate balance, that is synthesis is equal to breakdown. So level means normal, that is equal to 1 milligram per deciliter, right? But problem comes when synthesis is highly increased and breakdown is decreased, right? So ketone bodies level will increase, right? And when the levels increase in blood, it is known as ketonemia, right? The increased levels of ketone bodies in blood is known as ketonemia, and when the level further increased, the ketone bodies start appearing in urine, and it is known as ketonuria, right? So the level has increased in urine, it is known as ketonuria, and ketone urine. When the blood levels are increased in blood, it is known as ketonemia, and this common are known as ketosis. Both commonly are known as ketosis, right? And the typical feature of ketosis is acetone breath, right? And when you examine a patient, you can it can smell like a fruity smell, that is the acetone breath. When you will examine the patient and sit close to him, you will have fruity smell from his breath. And now I will discuss the metabolism in starvation, right? So what happens during starvation is that the principal source of energy is glucose, right? When glucose is utilized, then the fats are utilized. So fatty acid will break down to form acetyl CoA, right? So starvation, the glucose levels are depleted. The second source of energy is fats, and the fats are being broken down by beta oxidation to provide energy, and the product is acetyl CoA, right? And this acetyl CoA will enter TCA cycle, right? And during starvation, what happens is that this TCA cycle is impaired. Why impaired? Because the acetyl CoA levels are increased, but there is no sufficient level of xaloacetate to run the cycle properly, right? So this acetyl CoA is not further utilized and its levels further increase, and it is diverted for ketone body synthesis, right? Because acetyl CoA is precursor for synthesis of ketone bodies, right? Now I will discuss the metabolism diabetes mellitus. Now I will discuss the metabolism of ketone bodies in diabetes mellitus, right? There are two types, DM1 and DM2. In one, there is complete absence of insulin, and in two, there is insulin resistance, right? And so we can say that the carbohydrate metabolism is impaired and it is not going properly. So there will be increase in lipolysis, right? So when fats will break down via beta oxidation, what we will get high amounts of acetyl CoA, right? And this acetyl CoA is precursor for synthesis of ketone bodies, right? So there is great increase in production of ketone bodies, right? The normal level of ketone bodies is 1 milligram per deciliter in blood, right? So in severe uncontrolled diabetes mellitus, the level may increase up to 100 milligram per deciliter, right? And the urinary excretion can increase up to 500 milligram per day into urine, right? So due to impairment of carbohydrate metabolism, the lipolysis is increased, and lipolysis is increased there is increase in beta oxidation, and beta oxidation will lead to synthesis of high levels of acetyl CoA, right? Acetyl CoA is precursor for the synthesis of ketone bodies, so it will lead to ketosis, that is ketonemia and ketouria, and the normal levels was 1 milligram per deciliter, which may increase up to 100 milligram per deciliter, and the urinary excretion may increase up to 500 milligram per day. So that finishes with our metabolism in diabetes mellitus. For the regulation of the ketone bodies, do watch my upcoming video. So please like the channel and subscribe it, and please share with your friends. Thank you. Bye. God bless.